Right, hello everyone. Welcome to our afternoon session on sustainable seafood hosted by EIT Food. It's a real pleasure to welcome you all to our most important and crucial subject actually within the whole conference about sustainable seafood, which is an area that can really help resolve our climate challenges and food supply system over the next 20, 30 years. At the moment within aquaculture and a host of other areas, we see some significant damages and environmental challenges. And with our esteemed audience and panelists, I'm sure we're going to be tackle the latest innovations and products and companies that can really help resolve and create a far more sustainable seafood and aquaculture environment. Without further ado, please may I welcome Mercedes Grober, who's the Innovation Program Manager for the EIT Food team. Mercedes, please do take over. You are our esteemed moderator for the session. You'll be bringing everyone together and all our guests. So over to you, Mercedes, and enjoy. And I'll be hopefully lots of questions for the speakers today. So everyone get involved with as many questions as possible.
Thank you, Joe, for the introduction. Good afternoon, everyone, and welcome to this session on sustainable seafood. First of all, a big thank you for the organizers to inviting us today to talk about this important subject. My name is Mercedes Groba. I'm the Innovation Program Manager at EIT Food, where I lead the Sustainable Aquaculture area. And with me today, I have the pleasure to have three more panelists. I have Dr. David Aldrich from the University of Cambridge in the UK. I have Birger Orms-Marason from Matisse in Iceland. And I have also Olafur H. Fridtjofsson from Matisse in Iceland. Today I will have I will play two roles. I will be the moderator as well as the first speakers. Before starting, I'd like to remind all the participants that this is an interactive session. That means we would love to receive your questions. We will conduct the Q&A session at the end of all talks. So please add your question in the chat, and you don't forget to put the name of the speaker. During this special session from EIT Food, we will look at what EIT Food is, the innovation focus areas with special attention in sustainable aquaculture, our impact in 2019, and we will run into three EIT Food innovation projects and to know what are the results and progress. Let's start by understanding what EIT Food is and what we do. At EIT Food, our goal is to find solutions to the major social, environmental, and economic challenges that our food system is facing today. For example, we need to feed 10 billion people by 2050. We need to produce more food, but sustainably. In reality, some of us eat too much while the others don't have enough. The truth is, we are wasting food at a huge scale. To find solutions, we need all of you. We cannot do it alone. That's why we are bringing innovative players together to build a powerful community to transform the future of food. In other words, we want to create a food innovation ecosystem to make our food system more sustainable, healthy, and trusted. Most importantly, we want to ensure that everyone has access to it. And what are our strengths? Our partners represent over 85 leading agri-food companies, research institutes, and universities in Europe. We also bring together the best European agri-food startups and scales-up in our Rising Food Stars Association. Our work is carried out throughout Europe. Our headquarters is in Leuven, and we have 6 more regional offices to help us make an impact across Europe. And last but not least, we are all passionate about food. What's our focus? We have 6 strategic objectives. These strategic objectives focus on where we can make the biggest difference and where the greatest change is needed. For example, we aim to overcome low consumer trust, build a consumer-centric food system, educate the world, create healthier products, enhance sustainability, and catalyze food innovation. These are our 6 strategic objectives to transform the food system. We aim to improve the food system in 4 main ways: through education, with activities to attract, develop, and empower talent; through innovation, by fostering collaborations to develop innovative technologies, products, and services; through entrepreneurship, with activities to support entrepreneurs and startups across Europe; and through public engagement by involving people to become real change of the transformation. In 2019, to maximize our impact, we developed 6 strategic objectives, which are aligned with the UN SDGs, which refers Sustainable Development Goals. We launched these focus areas in January 2020, and since then, all our innovation projects are connected to them. Besides these 6 focus areas, we have also 2 transversal areas: consumer centricity, which continues to be the core of our business, and digital transformation, which plays a central role in the entire food chain. Now, I want to go deeper to explain our contribution through the sustainable aquaculture focus area. Let's start with a simple question. Do you know the seafood supply is in Europe insufficient? The reality is that we are currently relying on imports from many other countries, and the current systems come with a huge environmental cost. Now, by 2030, 2/3 of global seafood requirements could come from aquaculture. As seafood consumption increases, sustainable aquaculture must keep up with demand. Therefore, we must transform and expand our aquacultural practices into smarter circular systems that support our quest for our society. So, innovation is a must, as aquaculture comes with its own challenges. We have here several goals. Here. To provide healthy diets and improve nutritional value by increasing diversification and resilience of fish species, to improve conditions to increase aquaculture production without losing sustainability and nutritional value. And here we are talking about precision farming that includes 3 main areas: technological solutions to gather, analyze, and monitor data, the use of artificial intelligence, and robotics. All of these aim to increase operational efficiency. To increase consumer trust in aquaculture by improving food safety and traceability of aquaculture products, by finding alternative treatments, vaccines, and diagnostic tools, and by improving animal welfare and farm biosecurity. Another goal is address environmental challenges in 2 ways. One, we want to find alternative and sustainable raw materials for fish feed. And second, we want to decrease the impact on climate change and biodiversity through emerging production systems and new production locations. This includes on land as well as farther offshores. And last but not least, we want to involve local commitment and implementation of such solutions. Our integrated approach is unique for several reasons. First, it emphasizes transparency, integrations, and citizens. Second, it balances with new entrants to the system with established businesses. And third, it's aligned to or contributes to the UN SDGs as well as the European farm-to-fork strategy. For example, sustainable aquaculture contributes to SDG 2, which is ending hunger, SDG 3, which is ensuring healthy lives, SDG 12, which is ensuring sustainable consumption, SDG 13, which is taking urgent actions to combat climate change, and finally SDG 14, which is conserving and sustainably using oceans, seas, and marine resources. By adopting this approach, we will safeguard our environment through a more sustainable and circular food system. We will also deliver healthy dietary choices to our communities while we support the European economy. And in this last slide, I have included some numbers to show you our impact in 2019. We are happy to announce that it was a successful year. We reached out more than 2 million people online medium, 50,000 learners attending one of our educational projects, and we supported 127 startups globally. We are proud that 2019 was a great year with a great impact. And this year is even more meaningful. In response to the COVID-19 pandemic, we launched 2 additional initiatives. One, to support 13 agri-food startups, and the other to work on 13 innovation projects. So in closing, I would like to invite you to join us in growing the sustainable aquaculture area and in transforming the food system in Europe and across the world. And this is all from me. So thank you very much for your time and your attention. And if you would like to know more about EIT Food, please feel free to contact me. Once again, my name is Mercedes Groba, and I'll be happy to help you. So now moving to the next part of this session, we will present 3 EIT Food innovation projects within this area. And the first speaker is Dr. David Aldrich. Dr. David Aldrich is the senior lecturer in aquatic ecology, Department of Zoology, the University of Cambridge, and he will talk about the microcapsulated diet for sustainable aquaculture. Thank you, David, the floor is yours.
Thank you very much, Mercedes. So I'm going to talk about microencapsulated diets for sustainable bivalve production. And I should acknowledge from the outset our collaborators from ASTI in Spain, Rethink Resource in Switzerland, Mattis in Iceland, other colleagues at the University of Cambridge in the UK, and our commercial partners BioBullets in the UK. Now, as will be clear from the focus of this conference, today's food production systems and consumption threaten our health and our planet. And it's becoming increasingly clear that bivalves offer a nutrient-rich and sustainable source of food that can help us to meet global food security needs. So when we think of bivalves, we're particularly thinking about things like oysters and clams, scallops and mussels. So how good are bivalves as a food source? Well, this diagram here...
Sorry to interrupt you, David. We can't see your screen. Would you like me to share on your behalf?
Just a second. I do apologise.
Sorry, David, I wasn't sure.
Is that better?
Now, yes, I'm really sorry, David.
Oops, hold on. Is that okay? You can see that? Yep.
Perfect.
Perfect, thank you.
Okay. So So how good are bivalves? Well, if we look at this figure here, I've mapped a range of different potential food sources, widespread food sources, against a number of key nutritional components. So I've mapped omega-3, vitamin B12, zinc, selenium, vitamin A, iron and protein. And these show data for things like plants, crustacea, bivalves, and then other animal sources such as chicken, fish, and terrestrial meats. And the large blue polygon here shows that bivalves are pretty much the best food source across a whole range of these different nutritional components. And not only are bivalves great for food, they also have a way smaller environmental footprint than these other foods. So again, I've mapped against these same food groups some key environmental parameters: land use, freshwater use, eutrophication potential, and greenhouse gas emissions for a standardized amount of protein production. And the largest polygon there is the most environmentally harmful food group, and that's beef. And right in the middle of this diagram, that small bright blue polygon is for bivalves, showing just how small an environmental footprint bivalves have. So they're very nutritionally rich and they're very environmentally sustainable. And just to demonstrate how important bivalves could be globally, this is a figure that myself and one of my PhD students produced recently, which shows the market potential of bivalve farming across the tropics. And the red dots here show the areas that we've identified as viable for commercial bivalve production. And if we farmed just 1% of those potentially viable areas in the tropics, we could produce enough sustainable, high quality food to feed 1 billion people. And these little pale blue ovals show the amount of bivalve meat produced in these different regions in 2015. And these larger ovals, the number of people we could feed if we developed this potential market. And what's interesting is that certain geographies show real opportunities. For instance, Africa hardly eats bivalves at the moment, but we could really exploit this market. But to exploit this market, we need to increase production and demand across the whole value chain. So we need to look at the hatcheries, growing out of the bivalves, harvesting, storage and processing, distribution out into markets. And also we need to engage with consumers to make sure that they actually value bivalves as a food product. But one of the major bottlenecks in bivalve production is hatchery production. And one of the reasons for this is that bivalves have to be held in the hatchery for 1 or 2 years for most species before they are then large enough to grow out in the open ocean. And at the moment, there's limited supply of seed, of juveniles, and also the feeds available for growing up these bivalves are rather limited. And this is where we come in with our innovations in encapsulated diets. So if we look at the conventional way of growing these juveniles in hatcheries, they're grown typically on live algal cultures, as shown in this photograph. But live algal cultures have a large physical footprint in a hatchery. They're very expensive to maintain, and they're very vulnerable to bacterial contamination, which results in the whole bacterial culture being, the whole algal culture being lost. And what we do is produce dry microencapsulated diets, and these are cheap, they're easy to use, they can be tailored to meet the nutritional requirements of the organisms you're growing, and they're entirely reliable. So you just have a dry powder you can dose when you need it. So here is some quantification of how good microencapsulated diets can be. So in this figure on the left, we've got 3 values of to quantify how costly and damaging different diets can be, looking at carbon dioxide emissions, the cost of production, and the energy footprint. And the large red triangle shows typical hatchery algae, which is much more unsustainable than photobioreactor grown algae, shown in green, which in turn is less sustainable than our blue microencapsulated diets shown there. And if we look on the right hand side of this figure, The key things with our diets are that we source the nutrients from sustainably sourced sources, particularly algae. We encapsulate those products in a lipid coating, and these particles are optimized for their buoyancy, their shape, and their palatability to maximize their uptake by the bivalves. So the key objectives of our project with EIT Food are to firstly identify sustainable side streams for encapsulation, then to optimize the packaging of these products so that they can be used in a commercial setting, ideally with at least 6 months of shelf life under ambient conditions. We are then looking at commercial scale production of spat. We're working with consumers through sensory evaluations to show that they are indeed acceptable if we feed bivalves on our products. And ultimately, we wish to show commercial viability. So let's look at some of these steps in a bit more detail. So as far as the side streams go, we've identified a number of potential side streams. The two most exciting ones for us are algal side streams, where we have these products growing essentially as wastes in existing aquaculture systems. So we're taking other wastes from aquaculture systems, and through a circular economy, we're moving them into our products to grow sustainable bivalves. We're also looking at taking fish oils from waste streams in traditional fish processing. All these products can be encapsulated in our materials. The next thing we're doing is looking at optimized packaging, and here we've been working with commercial partners to produce fully scaled up products. And so we've been testing 3 different types of packaging in accelerated storage trials, and then we're testing our products for their shape, their buoyancy, their breakdown rate, surface structure, and particle size. And the key finding here is that we have indeed identified suitable packaging which allows our products to be supplied to the end user in a high quality, safe environment where they can last at least 6 months just sat on the shelf. Commercial scale production is obviously a key factor here. Now, we've already demonstrated that we can grow juveniles and spat in the laboratory. These are data from some work we did in the laboratory on European oysters. So along the bottom axis here we have time, and on the y-axis we have the juvenile size, the growth rate, if you like, of the European flat oysters. And these different graphs show different feed conditions. So the bottom 2 lines are for controls, where they weren't fed. The green line shows that with conventional algae we do get growth, but the purple line shows that with our diets we actually get greater growth at laboratory scales. For this project, we've been looking at scaled out production in full commercial settings in Spain. And actually, importantly, the EIT Food grant has leveraged additional support from the Assemble Plus program to allow us to really scale up these trials. And in this case, we've been working on the marine mussel, Mytilus galloprovincialis. And I'll only show some of the data here, but the results are really encouraging. So the first graph here shows 3 different treatments. NC is the no food control. A is where we fed the mussels just commercial algae. BB is where we fed them our microencapsulated diets, what we call biobullets. And then the ABB treatment is where we use a combination of commercial algae and our biobullets. And this first graph shows tissue growth over the run of the trial. And you can see that with the biobullets, we get just, or the microencapsulated biobullets, we get just as much growth as we do with algae. So that shows that our dry product is already as good as competing alternatives, and we already know that our products are cheaper and have a smaller environmental footprint. If we look at another measure of the bivalves produced in these trials, along the bottom axis here we have the same experimental treatments, but this time we're looking at body conditions, so the quality of the meat in the bivalves. And this shows that actually the biobullets, the encapsulated products, are getting really higher quality bivalves produced than those that were fed just with algae. So this is really exciting in this commercial scale setting, because it's showing that encapsulated diets offer an alternative to commercial algae, and even better, they may provide faster growing and higher quality bivalves. which is a great result. Well, are mussels produced with our products acceptable to consumers? But it's really important that we work with consumer co-creation in our products, and we've been doing a number of consumer testing trials. We've got some more underway at the moment, but this graph on the right shows comparisons of bivalves that have been fed either with our biobullets, the red line, a combination of algae and biobullets, or compared with commercially standard bivalves from a shop. And what's shown here is very clearly that independent consumers see no difference in the bivalves produced using our products than they do with standard bivalves for texture, taste, odor, and appearance. So our products are perfectly acceptable to consumers, which is a fantastic result again. And so the ultimate aim of this project is to demonstrate commercial viability. That's still going on as we draw all the components of this project together. But what I can say at this stage is that we have a scalable product. Everything looks great. It's working very, very well indeed. And so it looks like we are indeed commercially viable. So what are our conclusions from this project? Well, I hope I've demonstrated that microencapsulated diets could help to drive a step change in the commercial development of bivalve aquaculture, and that our novel technology offers a sustainable solution to helping to meet global food security. So thank you very much for your attention, and thank you EIT Food for supporting this exciting project. Mercedes, you're on mute.
Sorry, thank you. Thank you, David, for presenting to all of us and the audience your innovative products, and also to see how you involve consumers through co-creation in this project. Thank you so much for that. So the next speaker is Birger on Amazon. He's the project manager at Matisse. Matisse is the leading food and biotech innovation center in Iceland. He will present the Energy to Feed project. So Birger, the floor is yours.
Thank you, Mercedes. So can you see my screen?
Yeah.
Yes, it's perfect.
Okay,
yeah,
excellent. Thank you. All right.
Yeah.
So hello, everyone. My name is Birgir, and I research sustainable food at Matisse, Icelandic Food and Biotech R&D. And today I'm going to present the very relevant and exciting project called Energy to Feed, funded, of course, by EIT Food, where we are demonstrating a sustainable microalgae production intended for use in aquaculture feed. So a bit of a background, the aquaculture industry and salmonid production in particular maybe needs a sustainable and stable supply of feed resource materials in sufficient quantities and quality for future growth, in particular high quality proteins and the fatty acids EPA and DHA mainly, which so far have been ensured by inclusion of fish oil in the diets. About 70% of the available fish oil is currently being used in aquafeeds, but the global supply of fish oil is limited, and emerging omega-3 markets are competing with the aquaculture industry for this finite resource. So new sustainable sources of EPA and DHA must be fully developed to cover the global demand. And we think marine microalgae, they are regarded as promising alternatives as the primary producers of all the EPA and DHA in marine food webs. And in addition, about 60–70% of proteins used for animal feeds are imported to the EU. So EU is not self-sufficient when it comes to protein production. This large portion of the current protein demand is met with this import, especially concerning feed proteins. And this deficit I mentioned has been fluctuating, protein deficit has been fluctuating between or around 70% in the past 40 years, with, of course, severe concerns regarding food security and the general competitiveness of the EU. So it is therefore of vital importance to find sustainable protein and lipid sources that can be economically produced in quantities that meet the growing food and feed industry demand. And a solution we present here, which is WAXA's omega-3 rich microalgae production platform, addresses the main challenges preventing algal-based omega-3 to be used in aquafeeds. At scale, it presents a viable option for omega-3 enrichment and feed proteins, and is setting a new sustainability performance bar. So, Energy to Feed is a unique project around developing WAXA's technologies into sustainable sources of protein and lipid from microalgae, cultivated using geothermal power and natural CO2. So the project will demonstrate a truly sustainable microalgae production within a controlled environment for consistent year-round product quality, which will provide the European aquaculture sector with the next generation alternative to sustainably support its future growth. And in the consortium, we have, together with Matisse and WAXA, we have Siemens, who are developing automation technology in the project, for example, with WAXA, and Waitrose, who are looking into the market and business sides of things. So what is so special about this anyway? Well, I think for starters, the microalgae plant, WAXA's microalgae plant, is located next to one of the biggest geothermal power plants in the world, near the capital area of Iceland, in Hellisheiði. The microalgae production uses carbon dioxide from the power plant as a carbon source for algae growth in vertical photobioreactors using LEDs, and also utilizing excess hot and cold water from the power plant for cooling and downstream processing, for example. And here we can see actually a photo of the geothermal power plant in Hellisheiði, and WAXA's production plant is just below this picture here. And the top image is from inside WAXA's production platform. So continuing on that, WAXA is currently claiming, backed up by initial research, that they have a carbon negative production of microalgae and tiny fraction of the land use compared to other types of microalgae production methods, and not to mention other types of common protein and omega-3 production, and very minimal water use. And microalgae, I mean, they are the primary producers of EPA and DHA, as I mentioned, that accumulate over the trophic levels in the marine food webs. Most are photosynthetic, but unlike plants, they do not have stem, roots, and leaves, which allows up to 10 times more efficient mass transfer and growth compared to terrestrial plants. And microalgae are therefore considered a very promising feedstock for future sustainable supply of protein and lipids for commodities and specialties for actually both food and non-food products, such as feed. So our main aims with this project, I mean, they are to essentially pave the way to commercial scaling by demonstrating a modular production technology, converting clean energy to sustainable aquafeed, including an environmental and techno-economic analysis of the entire production to end user chain. The modularity, controllability, and automation of the design, which is being developed by Siemens with WAXA, should be proven and optimized in this activity, which is the key to lowering scale-up risks, as well as supporting CapEx economies of scale. So this modular technology can be adapted to other industrial areas which have excess heat and CO2 sources. And secondly, the developed algae product used as an ingredient in aquaculture diets and as an alternative to fish meal and oil and other commonly used ingredients. So which is then what we have been and are currently doing right now. And here we can see some initial results from just ended study on Atlantic salmon feeding, where we have developed feed containing algae from Vaxa and compared it to an industry standard feed as a control. So the first table to the left shows the nutritional composition of the algae used for this specific study. And in the table to the right, the fatty acid composition of the two diets. So we actually in this trial we were focusing a bit more on the fatty acids. And then in the table at the bottom, we see the fatty acid composition of the Atlantic salmon itself after feeding for about 6 weeks, and the significant difference in especially EPA and omega-7 increase, but also some for DHA. So we can see that EPA 49% increase, and omega-7 49% increase, and 4% increase in DHA. So this particular strain used is very high in EPA, but it can also be combined with another strain, for example, in the feed, who is higher in DHA to balance this out. So this is actually very, very promising. And here we can just see some photos from the trial of the Atlantic salmon, roughly, I would believe, around 300 grams in this photo, and where we are putting this algae feed on the feed belt. So, since it's it's great project, we think, with many things ongoing, and since we don't have too much time to go over this here, I only showed you a very general overview of the project and the results so far. We are working on completing a lot of work right now, and we really look forward to share the content of this project with everyone very soon. And to conclude, we think that Energy to Feed presents a sustainable platform to produce high quality microalgae rich with omega-3 and protein. And this provides a sustainable choice in aquaculture feeding. And as we believe, demonstrated, Vaxa's microalgae can be formulated in salmon feed to significantly boost its omega-3 content. Yeah, so that's it for me now. And finally, I just want to thank the partners in the project, and of course EIT Food for funding this and making this happen. Thank you.
Thank you, Birgit, for this interesting talk and the great project. It's a project that will contribute to the sustainable, to make our food system more sustainable, for sure. Thank you so much. So moving to the last talk of this session, we have with us Olafur H. Fridtjonsson. He's the research group leader in biotechnology at Matís in Iceland, and he will talk about the project called the Fermented Seaweed-based novel feed Additives. So the floor is yours.
Yeah, thank you. I'm going to share my screen now. So hopefully you see my screen.
Yes, we can see it.
Okay, fine. So to start with, I want to thank you for this opportunity to introduce the EIT project SEAFEED and its results. My name is Olafur Fridtjonsson, and I come from Matís, as Birgir, who spoke here before me. So with expanding aquaculture, the request for alternative aquaculture feed is increasing. So the need for alternative proteins in aquaculture replacing fish meal or soya meal is evident. And Martis has been very active in exploring ways to develop new sources or proteins in feed. Now, this is important as moving to alternative feed will have significant positive environmental and economic impact. For example, by valorization of underutilized resources. And Birgir, you reviewed this very well here before. So we have focused on seaweed as feed additive, not only seaweed but fermented seaweed. Now, fermentation is a good way of preserving seaweed and increasing its nutritional value. So I'm going to introduce to you some of the results of the SEAFEED project supported by EIT Food, and where the aim was to develop novel aquaculture feed formulation based on fermented seaweed with as a potential alternative to antibiotics. Now, we are not replacing feed proteins. Rather, we are including health-promoting bioactive ingredients, prebiotics from seaweed, and probiotic bacteria beneficial to the gut microbiome. Now, seaweed biomass is largely underexploited feedstock in Europe, and biorefining applications are at an early stage. So by using seaweed, we are contributing to valorization of sustainable underutilized feed resource in Iceland and hopefully Europe in the future, and we are using locally produced ingredients. So in recent years, seaweed has been studied with regard to its composition and bioactivity. And before this project started, we already had studied different types of seaweed regarding the potential as health-promoting supplements in feed. For this project, we selected 2 brown seaweed species as feedstock, Laminaria digitata and Saccharina latissima. Now, these brown algae species are abundant source in Icelandic coasts, and they are known to be especially rich in carbohydrates, including mannitol and laminarin, which are reserved carbohydrates. But due to the high carbohydrate content, they should be an ideal fermentation feedstock. Now, fermenting seaweed and utilizing it as feed is not a new idea. There are old reports of farmers in Iceland living close to the sea, the coastline, who ensilaged seaweed and used it as feed supplement for sheep and also as fertilizers. Currently, there's a company in Denmark which produces feed with fermented seaweed mixed with rapeseed meal, and this feed is for porcine and poultry. But here we wanted to develop it for aquaculture. Now, to go a little bit, tell a little bit about the process. We start with dried and milled seaweed, sustainably harvested around Iceland, and dried. It is dried using cheap geothermal energy. We pre-process the seaweed by heating to release carbohydrates from seaweed, and also to make it available to the fermenting bacteria. Also to reduce the native microbial flora. After pre-processing, we start the fermentation by adding starter cultures, and the fermentation goes on for a few days at semi-anaerobic conditions. Now, after the fermentation, the pH drops, of course, and the lactobacillus species dominate the seaweed slurry, as the results of the microbiome sequencing shown here on the slide indicate. The control incubation made without the starter culture In control incubations made without starter culture, the pH remains stable usually, and the slurry contains mostly marine bacteria and some other environmental spore-forming bacteria. On this slide, we see that during fermentation, mannitol and free glucose released by the pre-processing are used by the fermentation strain. On the other hand, after fermentation, when mono and disaccharides have been used, the bound glucose in the laminarin polysaccharide and the laminarin oligosaccharides remain in the slurry, as we see here in the chromatograms on the right. Now, this is good because these oligosaccharides serve as potential prebiotics. Now, marine resources are often high in trace elements such as cadmium and also high in arsenic and iodine. Therefore, it's essential to monitor the content of these elements in the feed formulation. And as the table shows here, the content of arsenic, cadmium, and mercury is not increased by inclusion of the seaweed. However, the iodine content is increased and even exceeds maximum levels according to EU regulations. But we learned and found out, however, that this can be easily controlled and reduced by simple balancing pre-steps before the pre-processing. Now, the fermented seaweed was tested as feed additive for salmon in aquaculture. Each seaweed type was added to complete feed as 4% inclusion, and the same feed without seaweed was used as control. At the start of the trial, the fish was around 500 grams. It was adapted for 10 days and then fed for 75 days on the experimental feed and control feed. Now, if you look at the results, the growth average values, the specific growth rate or the weight increase of laminaria-fed salmon was a little bit higher than the control group, while the growth of the saccharina feed was a little bit lower. Now, the contagion factor or the relationship between weight and length of the fish is equivalent for the groups and obviously increased during the study, indicating that the fish is in good health and feeding well. Now, when the fish was slaughtered, the gut content from number of individuals from all groups was collected, and a microbiome analysis by 16S RNA metabarcode sequencing was carried out. Unfortunately, the COVID situation delayed this work a bit, and we are still waiting for the final results. But we had the opportunity to prepare a meal at the end of this day, and as you see on the picture down right, and the salmon tasted delicious. But of course, proper sensory analysis was carried out at the Mattis laboratory by well-trained sensory personnel, and also a small consumer survey was carried out. But also here, the COVID situation had an impact. A lower number of participants were involved than originally planned, yet enough to draw some conclusions. The general observation from both the sensory panel and the consumer survey was that no significant difference was detected in flavor, odor, or texture between samples, the fish fed on seaweed feed and the fish fed on control. Now we measured also the color and the content of protein, fat, and water, and there was no significant difference between any of the groups in any of those assays. Now, as previously mentioned, the concentration of trace elements, lead, cadmium, mercury, was similar in the seaweed feed as in the control feed. However, the iodine concentration in the seaweed feed was significantly higher compared to the control, as expected, and therefore the iodine content in the salmon fillets from the experimental groups and the control was analyzed applying accredited methods. Now the results are shown here, both in a chart and in a table. Now, as evident from the chart, the high iodine content of both seaweed types is reflected in the feed, and the iodine is clearly transferred to the salmon product. Now, the left side of the table here shows the measured values in salmon muscle in micrograms per 100 grams. Now, right side of the table shows the amount of iodine found in 200-gram portion of the salmon, with recommended and daily intake of iodine for adult here in the middle of the table. Now, these values are from the Commission recommendation from 2018. Now, based on these values, for example, for Laminaria digitata, a 200-gram portion of salmon would provide approximately half of the daily recommendation of iodine for an adult. And so this may be quite good, because we know that iodine deficiency is the cause of many health problems. So I'll just here at the end to summarize results. The seaweed fermentation process developed is simple and effective. The fermented seaweed prevents growth of fouling microbes, or the fermentation prevents growth of fouling microbes, and it increases the shelf life of the product. Mannitol and free glucose is consumed, whereas bioactive oligosaccharides are left intact, and that's good, because these are the prebiotic compounds. Now, the fish liked the feed. It had a healthy appearance, and growth rate was high, comparable to applying high-quality fish meal feed. No significant difference in odor, flavor, and texture was detected according to sensory analysis and survey. Now, iodine levels in the feed is high, and it's transferred to the salmon. A single serving of seaweed-fed salmon can provide a half of the daily recommended iodine intake of an adult. So currently we are evaluating the exploitation potentials of this product with the beneficial partners of the project, but there are still some open questions which need to be addressed. So we will continue to work on this for a while, I think so. So yeah, I just want to thank EIT for the EIT Food for the support, funding, and our collaborators, that was the University of Helsinki and Quadram Institute, for their collaboration. Thank you.
Thank you. Thank you very much. I think we have a couple of minutes for questions, and I received, in fact, 3 questions for one for each. So let's start with the first question is for Berger. And the question is, you mentioned something about that the technology is you will be able to use in another industrial areas. So you give us a little bit more details about this?
Yeah, we lost you slightly during that question, Mercedes,
yes,
but
let's repeat. So the question is, the technology, the sustainable micro algae technology that you are developing, you mentioned that it will be able to use in another industrial areas. So could you please give us a little bit more information about this?
Yes, of course. So within bivalves, we have already demonstrated that we can also use it to improve the quality of brood stock. Actually, that's another component of this project I didn't have time to cover. And we've actually already demonstrated significant uplift in brood stock quality. These feeds also have potential for being used in shrimp production systems and fish production systems. We haven't really looked at that in any detail. And the other thing we can also do that we've had some good preliminary results with is that it's a platform technology and we can encapsulate anything we like. And so we've also been looking at fortification of bivalves in the depuration stage to uplift the quality of certain vitamins like vitamin A and D, and also omega-3. And we can even use the technology to uplift particular flavors that consumers might particularly like within the product. So it's a very, very expandable platform technology.
Thank you so much. And now, Birgit, for you, the question is, are you involving consumer co-creation in your projects? If so, in which way, what you are doing with the consumer and citizens to develop your technology?
For me, actually, in this project, we are actually not involving consumers or stakeholders, to be honest. So we decided from the start that so this would be an initial run of the technology and testing it. So for any future projects and plans for this, the next steps would for sure be to involve interested stakeholders and, of course, the consumers.
Thank you. And for the question for you is, why do you have you choose salmon to do the trial and not another species? And are you planning to extend the trials to another species?
Well, we had this connection to this cultivation facility, and which made it easy to use salmon, and we are used to use salmon in our aquaculture experiments. Now we can easily test this on trout as well, char, here in Iceland. But we would, for other species, we would probably have to collaborate with partners abroad.
Thank you. Well, and a friend, it's already time to close the session, so we don't have time for more questions, but for sure, everyone wants to know more about this EIT Food project or EIT Food. Please contact us and we are happy to answer any question.
Thank you, Mercedes. I would like to also extend my appreciation to all the presenters today in the Sustainable Seafood session. Thank you, David, Birgir, Olafa, and Mercedes for your excellent moderating. That was a really riveting session. I hope we can all work together to support all the good goals that the EIT are working and funding. So thank you all. And also thank you to everyone who's watched the Sustainable Foods Conference over the past 3 days. We've had a real wonderful audience and participation. Do remember that the event is still open for networking and to rewatch content for the next couple of weeks and months. So do get in touch with everyone on board in this session for more information. I'm sure they'll be happy to get in touch. So thank you all, and we'll hope to see you next year once hopefully COVID is behind us. So take care, everyone.